Here we have a DNA strand that is ready to replicate. Some structural components that factor in to DNA replication is the fact that it's a double helix shape and the two strands run anti-parallel of each other. DNA replication begins at the origin of replication. This forms a replication bubble, and at each end of the bubble, the replication fork is where parent strands are being unwound by DNA helicase. Here we have our replication bubble. As you can see, the red strands on the outside represent the deoxyribose sugar and the phosphate group backbone. These are held together by covalent bonds and are very strong, cannot be broken during DNA replication. In the middle, we have our nitrogenous bases, purines and pyrimidines. Purines go with pyrimidines in a concept called complementary base pairing and are held together by hydrogen bonds that are much weaker than covalent bonds and are split during DNA replication. To make this replication bubble possible, there are multiple components that are helping out helicase. For example, topoisomerase works ahead of the fork to relieve tension on the DNA, breaking and untwisting. Also, the single-strand binding proteins on the outside help make sure that hydrogen bonds do not free-form between the complementary bases. DNA is read from the 3' end to the 5' end. However, it is built from the 5' end to the 3' end. Since DNA is anti-parallel, RNA primase works in opposite directions to lay down primer. This causes a lagging and leading strand. The leading strand is working towards the 5' end to the 3' end, and the replication fork is at the 3' end of the strand. However, the lagging strand also works from the 5' end to the 3' end. However, the 5' end is where the replication fork is. Since the lagging strand works away from the replication fork, this causes the primase to jump back after you opening the replication fork occurs. This causes Ozaki fragment. Step DNA polymerase III adds free nucleotides into the template, elongating the new stranded DNA by adding complementary base pairs. And DNA polymerase I removes the primer and RNA and adds DNA back in. The final component is DNA ligase that comes in and reattaches all the fragments together, creating two new separate strands. Each strand contains one of the parent strands.